Squeakers, huddle in. We're about to test a long-held assumption that life began simple and only later became complex. Yet the earliest known cells like Methanobacterium and Cyanobacteria defy that cozy tale. Far from being primitive blobs, they contain an intricate web of molecular machinery that operates like a cosmic symphony. Methanobacterium is an ancient, oxygen-hating microbe that cheerfully gobbles hydrogen and carbon dioxide, farting out methane as a byproduct. Think of it as a microscopic cosmic chef, cooking methane soufflés in swamps, cow bellies, and volcanic vents. Picture Methanobacterium as the supermouse of microbes, thriving in scorching hydrothermal vents, extreme acidity, or buried deep in the Earth's crust. It doesn't just survive, it laughs at harsh conditions and generates methane like it's a cosmic gas factory. Cyanobacteria are ancient microscopic maestros, the original solar-powered superstars. They harnessed sunlight, water, and carbon dioxide, releasing oxygen into our planet. They were harnessing sunlight more efficiently than our best solar tech. Talk about supercells. In Darwin's day, limited technology made cells look like fuzzy blobs of protoplasm. He naturally focused on how tiny changes over vast time could lead to complex organisms, without realizing the cell itself was already a molecular marvel. Today's research reveals these so-called simple cells are anything but. They're packed with sophisticated components and a near-universal genetic code ready to orchestrate astounding diversity. This revelation forces us to refine the old notion of time plus random changes, because the very foundation of life was never truly simple. It just appeared that way before modern microscopy and molecular biology. Yet in truth, the so-called simple cell is anything but. It's already bristling with outrageous molecular genius. All life's dazzling variety emerges by rearranging that foundationally complex system. If these supposedly primitive cells already display extraordinary sophistication, one wonders how we even define advanced. They were never mere blobs. Modern research shows they were equipped with error correction, energy turbines, and coded instructions right from day one. So now, dear Squeakers, we'll embark on a series of checkpoints to reveal exactly how jaw-droppingly precise even the earliest cells are. Is this mind-bending complexity merely a fluke of chemistry, or the unmistakable fingerprint of something far greater? Prepare yourselves. What passes for primitive at first glance may well hide a universe of design from day one. With each checkpoint we explore, ask yourselves, could this staggering complexity really be just a cosmic accident, or is it the unmistakable fingerprint of a masterful creator? Checkpoint 1. The magnificent 20 amino acids. Life's proteins are all built from a set of exactly 20 amino acids. Why not 5 or 15 or 37? This particular group strikes a Goldilocks balance, covering water-loving, oily, acidic, basic, bulky, tiny, and everything in between. It's as if nature had to select just the right notes for an infinite molecular symphony. One misstep, say missing a crucial side chain, and you'd risk biochemical chaos. Yet from archaea to mouse to human cells, everyone sticks to the same 20 puzzle pieces. Note, while most proteins use the same core set of 20 amino acids, certain organisms incorporate 1 or 2 additional amino acids, selenocysteine and pyrrolysine, bringing the total to 21 or 22 in rare cases. 1. Collecting the pieces. Amino acids don't just float around in tidy beakers. They're tricky to form, easily degraded, and come mixed with random byproducts. Before you can even build proteins, you'd need a stable supply of these 20 purified amino acids. No extras, no weird isomers. Origin of life experiments, like Miller-Urey, might yield a handful of amino acids, often left and right-handed forms, plus gunky tar. Never the full curated set. How do they magically purify themselves in harsh conditions? There's no lab technician out in the wild cleaning up the sludge. 2. Hooking them up into polymers. Even if you gathered 20 perfect amino acids, you must now chain them together via peptide bonds. In water, such condensation reactions are notoriously difficult. Water loves to break those bonds. In living cells, elaborate enzymes and ribosomes orchestrate each peptide bond with precision. By chance alone, you'd expect a random soup, not carefully sequenced proteins. Worse, random hooking of 20 amino acids in any order gives you an astronomical number of potential sequences. Over 100 combinations for a 100 amino acid protein, way beyond what random collisions can systematically test. 3. Why not 2, 10, or 40 amino acids? Suppose natural processes started with just 2 or 3 amino acids. You'd get severely limited protein shapes, hardly enough diversity for metabolism, regulation, or structural components. Even if 10 amino acids worked, how did the other 10 seamlessly appear and integrate later? Why didn't life end up with 45 or 87 if each new amino acid could spontaneously evolve in? No experiment shows that random chemical soups converge on exactly 20 universally. The same puzzle arises if you consider what each amino acid does in protein folding. The existing set is finely tuned. Some are hydrophobic, hide from water. Some hydrophilic, love water. Some form disulfide bridges, etc. It's a perfect chemical palette that's extremely tricky to expand or reduce without widespread dysfunction. Number 4. Futile random experiments. People sometimes imagine nature trying endless experiments over millions of years. But raw trial and error in the real environment leads mostly to the degradation of fragile molecules, especially if no protective lab conditions exist. Heat, UV radiation, pH swings, you name it. It's all hostile. Each partial success would likely be erased before forming a stable functional set. It's not as though nature can systematically log attempts and refine them like a skilled researcher. The environment is too chaotic and ephemeral. Why this points to a fingerprint of God. The consistency across all life, from ancient microbes to modern mammals, can't be explained by random, incremental steps with no guiding intelligence. Instead, it's like someone handpicked these 20 specific building blocks from the start, ensuring the maximum range of biochemical possibilities while maintaining coherence. Imagine discovering a precisely tuned orchestra of exactly 20 unique instruments, each perfectly selected for its specific timbre. No duplicates, no extraneous instruments, pure harmony. The synergy is just too perfect to be accidental. If we can't even replicate this selection artificially in a lab without enormous effort and advanced technology, how can we claim a random mud puddle achieved it long ago? Hence, dear Squeakers, the 20 amino acid system screams design. A mishmash of leftover chemicals doesn't freeze into such a balanced set, nor do they conveniently self-assemble into robust polymers. The deeper you probe this perfect palette, the more it resembles a hallmark of a masterful designer, rather than a haphazard mishap of nature. Checkpoint number 2. Ribose, the fragile sugar backbone. Ribose should degrade easily under heat or chemical stress. Logic says it wouldn't last long in the turbulent early Earth. Yet in cells, be they archaea or bacteria, RNA with ribose endures, ferrying genetic instructions. Picture balancing a delicate glass ornament amid raging storms, and it never shatters. That's the improbable feat early life pulled off. Ribose is a delicate 5-carbon sugar that forms the backbone of RNA, that all-important courier of genetic instructions. Outside a living cell, forming ribose from scratch is extremely challenging. The foremost reaction often cited by origin-of-life researchers can produce sugars, but also a mess of tar, random isomers, and destructive byproducts. And if you do manage to isolate ribose, it quickly reverts to useless fragments unless carefully protected. Nature has no lab technician. So how do you run a multi-step purification under scalding heat, UV bombardment, fluctuating pH, and countless side reactions? At the same time the environment is supposedly generating pure ribose, it also needs to produce the magnificent 20 amino acids, stable phosphates, and nitrogenous bases to form nucleotides. Are we to believe multiple improbable chemistries were all working flawlessly in parallel on a chaotic primordial Earth? If you think time solves all, ask yourself, does waiting millions of years magically filter out the tar and preserve fragile sugars? Or is time actually the enemy, leading to more breakdown and more contamination? In living cells, even ancient ones, ribose is formed and immediately integrated into RNA by specialized enzymes. The cell is like a miniature laboratory, controlling temperature, pH, and reaction steps. Outside in open water or on a hot volcanic surface, ribose would degrade in no time, forming goo rather than neat nucleotides. So how did lifeless nature run a perfect ribose assembly line with no cell yet to safeguard it? Origin-of-life experiments like Miller-Urey or Formose typically generate small molecules, some amino acids or simple sugars. Headlines proclaim, "We made the building blocks," implying life's origin is around the corner. But discovering a few random needles doesn't mean a whole watch emerges spontaneously. Yes, we may form ribose occasionally in a lab flask, but it's heavily controlled and still ephemeral. Translating that into stable RNA fully harnessed by a cell is a colossal leap. The deeper we dive, the clearer it appears that natural processes in an untamed environment can't orchestrate ribose's delicate role in information transfer. Hence, dear Squeakers, ribose is no trivial sugar. It's the fragile backbone of RNA, stabilized daily by living cells, yet ironically impossible to maintain in raw nature for long. Whenever origin of life researchers claim, "We formed a bit of sugar in the lab. See, life's on its way," remember, those are just random needles, nowhere near a functional watch. The improbability of harnessing ribose, amid countless destructive factors, stands as a bold fingerprint of God, revealing a profound intelligence behind life's foundational chemistry. Checkpoint number 3, phosphate, nature's superglue. Phosphate, po qui mance, is like a hyper-energetic chemical hugger. It loves bonding with just about anything in sight. If left uncontrolled, you'd get useless clumps and precipitates, not the elegant structures essential for life. Yet in cells, phosphate is precisely harnessed to build DNA-RNA backbones, power ATP, and even form phospholipids in membranes. It's as if someone trained superglue to stick only where needed, with no chaotic side mess. Another glaring hint of directed engineering. Number 1. What's phosphate got to do with amino acids and ribose? Ribose needs phosphate to form nucleotides, nucleoside phosphate nucleotide, the building blocks of RNA and DNA. Without that phosphate link, you don't get stable genetic polymers to store or transfer instructions. Amino acids ultimately depend on ATP, the phosphate-packed energy currency, to power their linkage into proteins. Every time your ribosome forms a peptide bond, it uses energy from ATP or GTP. Both rely on phosphate. Cell membranes often have phospholipids providing structural integrity. The phosphate head is hydrophilic, while the fatty tails are hydrophobic, creating a stable bilayer for the cell boundary. 2. Magic behind phosphate bonds. A phosphodiester bond holds together the sugar-phosphate backbone of DNA, RNA. This bond is stable enough to keep genetic information intact, yet breakable by specific enzymes when needed, e.g., replication or repair. High-energy phosphate bonds, as in ATP, store and release energy in a controlled manner. ATP is like a tiny rechargeable battery that's constantly powering life's chemical reactions. Without precise phosphorylation steps, you couldn't activate sugars, amino acids, or other molecules for biosynthesis. Phosphate is the master enabler of countless metabolic pathways. 3. The paradox origin of life theorists must explain how phosphate, typically locked up in insoluble minerals or sequestered in apatite, became concentrated and activated in the wild. Under random conditions, it loves reacting indiscriminately, forming insoluble clumps with calcium, magnesium, or iron. In a cell, specialized enzymes channel phosphate exactly where it's needed, whether assembling a DNA strand or loading ATP. But these enzymes themselves need ATP for energy and are coded by DNA. That's a circular puzzle. You need phosphate-based molecules like ATP to build the enzymes, but you need those enzymes to process phosphate in the first place. Number 4. How the cell handles phosphate versus raw nature. In cells, strict control. Dedicated transport proteins move phosphate ions across membranes. Enzymes precisely attach or remove phosphate groups from substrates. No random precipitation clogs the system. It's akin to having a carefully managed superglue gun in a high-tech factory, dispensing adhesive only when and where required. In raw nature, phosphate often ends up in insoluble mineral deposits, especially if you mix it with metal ions in a chaotic environment. No built-in system knows how to gather, dissolve, purify, and harness phosphate. The result? Little chance of forming stable, high-energy compounds or genetic backbones spontaneously. Number 5. Why this speaks of design. The synergy between phosphate, ribose, and amino acids highlights an intricate interlocking relationship. Phosphates fuel the creation of proteins, which in turn manage phosphate usage, while nucleotides require phosphate to exist at all. Claiming raw collisions sorted all this out in a scalding, chaotic early Earth environment feels more like wishful thinking than chemistry. The endless hype from some origin of life labs, we made a tiny bit of phosphate-based compound, ignores the fact that wild nature lacks the advanced labs, purification steps, or catalysts to replicate that success. Observing how elegantly and universally phosphate is deployed, powering ATP, stabilizing DNA, RNA, and building cell membranes, suggests more than blind accidents. It looks like coordinated design, a hallmark of an intelligent blueprint behind life's architecture. So, dear Squeakers, next time you see a DNA model or hear about ATP's mighty phosphate bonds, remember, this sticky hugger had to be tamed and orchestrated by something far smarter than chance. Another reason to suspect an intelligent craftsman set all this in motion. Checkpoint number 4, nitrogenous bases. Order from chaos. Let's now talk about nucleotides, the fundamental units of DNA and RNA. Think of them as tiny info packets whose mission is to store, transmit, and express all the data needed for a cell to function. Each nucleotide has 3 parts: a nitrogenous base, A, T, G, or U. This is the letter that helps encode vital genetic instructions. A sugar, ribose or deoxyribose, forming the structural backbone. A phosphate group, which links nucleotides into long chains, like beads on a string. Their task? The nitrogenous bases line up in a specific sequence, forming a code that tells the cell which proteins to build. Nucleotides form complementary pairs. A pairs with T or U, C pairs with G, letting cells replicate their genetic blueprint accurately. Once translated, that code becomes the thousands of unique proteins that give each cell its identity and abilities. Their vision? Universal communication. By using the same molecular alphabet, all living organisms, from archaea to mice to humans, can store and read genetic information with astonishing fidelity. Seamless integration. Nucleotides don't just float around alone. They also partner with sugars, phosphates, amino acids, and more to create an interconnected web of life's processes. A, T, C, G, or U in RNA, can form clumps or degrade in random conditions, far from the pristine sequences we see in biology. Yet within even Earth's earliest microbes, these bases line up like a perfect typed message, storing coherent genetic instructions. If there were no guiding principle, we'd expect nothing but molecular gibberish. Instead, what we find is a code so robust it has persisted across billions of years. God tech, as I like to call it, an unrivaled marvel of durability, precision, and universal appeal. Linking these nucleotides end-to-end flawlessly to create genetic code is like assembling entire novels from letters tossed in a hurricane, improbable without deliberate guidance. Consider the sheer scale. Methanobacterium's genome has about 1.7 million nucleotides. Cyanobacteria can exceed 3 million. Every single nucleotide must align perfectly, or vital information is lost. Paradoxes to ponder. Ribose is fragile. How did it survive to form nucleotides in the first place, especially with phosphates so prone to random bonding? Bases can degrade or form incorrect linkages if they're not guided. How did they consistently line up as neat base pairs, complete with complementary strands? Coded information. The biggest paradox. Chemical letters that carry meaningful instructions, not random babble. Where does that meaning come from in raw chemistry? You might have expected an early cell to be super simple, a vague blob. Instead, your head is probably spinning from the intricacies we've seen so far. Countless bits of chemistry must line up perfectly, again and again, for life's code to remain coherent. This complexity doesn't shrink under scrutiny. It grows more astonishing. Checkpoint number 5. Chirality and Gideon's Fleece. Well, let me introduce you to chirality, a fancy science word describing molecules that exist in 2 mirror image forms. Think of your paws, dear Squeakers. You have a left paw and a right paw, almost identical but not quite. That's chirality. Now here's the mind-bending twist. All proteins in living cells use only left-handed amino acids, while DNA and RNA use right-handed sugars. If nature were purely random, we'd see a messy mix of left and right, causing absolute chaos. Instead, across every known organism, it's left-handed amino acids and right-handed sugars, like 2 contradictory signs at once, all around the planet. This is as astonishing as the biblical story of Gideon's Fleece, where Gideon asked for 2 wildly improbable, opposite miracles to confirm his calling. Dew appeared only on the fleece, while the ground stayed dry. The next day, dew fell everywhere on the ground, yet the fleece was bone dry. Chirality is our modern Gideon's fleece. It defies normal expectation just as much. Amino acids come in left and right-handed forms, yet life picks only left. Sugars come in two mirror forms, yet life picks only the right. Origin of life researchers might say, oh, maybe left-handed amino acids appeared first, then tried the right, then a million years passed, and poof, a perfect arrangement. But remember, early Earth was harsh, scorching heat, radiation, fluctuating pH. Time was the enemy of these fragile molecules. There's no real natural selection for random chemical soups in such a frying pan. If left or right forms emerged, they'd likely degrade before they ever chose a single hand. So, dear Squeakers, the improbable alignment of only left-handed amino acids and only right-handed sugars across all life is like those two opposite do miracles, repeating in every cell. A mind-boggling improbability indeed, one that many say shouts no mere accident. Dear Squeakers, we've explored 5 compelling checkpoints highlighting paradoxes not easy to dismiss as random chance. As we summarize, let's reflect deeply on the mind and character of our Creator God revealed through these remarkable designs. Checkpoint 1. The Magnificent 20. Life's precise selection of exactly 20 amino acids from hundreds of possibilities is no accident. It's like an artist carefully choosing the perfect palette, not merely for immediate use, but thoughtfully designed for endless diversity and future adaptability. This intentional, future-proof design showcases God's extraordinary providence, ensuring life thrives sustainably through countless generations without ongoing intervention. Picture this. Even a seemingly primitive cell like Methanobacterium contains approximately 2 million protein molecules, each composed of about 350 amino acids, totaling roughly 700 million amino acids per cell. This staggering complexity mirrors God's instruction to Abraham: count the stars of the sky, an impossible task that highlights the Creator's boundless intentionality, meticulous planning, and abundant provision beyond human comprehension. Checkpoint 2. Ribose sugar is exceptionally fragile, yet critical for RNA structure, ATP energy transfer, and cellular signaling, CAM. Too delicate for natural processes alone to integrate into life. Checkpoint 3. Phosphate, nature's superglue. Wildly reactive phosphate behaves chaotically outside cells, yet within, it's precisely channeled for genetic backbones and energy, ATP. This extraordinary chemical discipline is as improbable as muddy gold purifying itself and assembling into a perfect ring over vast time spans. It demonstrates God's power to transform chaos into order and purpose. Checkpoint 4. Nitrogenous bases, ordered code. Bases A, T, C, G meticulously store coherent genetic information, remarkably stable across billions of years. Just as Jesus assured us that even the hairs on our heads are numbered, God intimately knows every molecule and every gene within even the simplest cell. Checkpoint 5. Chirality, Gideon's fleece. The consistent molecular handedness, left-handed amino acids, right-handed sugars, is biologically miraculous, paralleling Gideon's fleece miracle. Such precision invites each of us, like Gideon, to witness firsthand God's intentional and unmistakable fingerprint. God is clearly a God of order, demonstrating that with the right framework and design, life can flourish harmoniously for billions of years. These components, amino acids, ribose, phosphate, nitrogenous bases, and chirality, do not merely coexist. They profoundly depend on each other. Amino acids form proteins that manage phosphate, phosphate energizes these proteins, ribose forms the backbone of RNA, and nitrogenous bases store instructions to orchestrate the whole. This deep synergy isn't accidental but reflects the wisdom of a creator who meticulously ensures that every component works harmoniously for life's continuity and flourishing. Consider this: if these 5 checkpoints alone, just the raw materials of life, are already fantastically complex, imagine how much more complexity awaits. We haven't yet touched upon replication, error correction, or metabolic regulation. Yes, scientists diligently explore countless chemical pathways, striving to discover life's blueprint. Yet each incremental step reveals how immense the challenge truly remains. We'll explore more fascinating checkpoints in future episodes. But for now, dear Squeakers, let's call for cheesecake. After all, absorbing such awe-inspiring wonders deserves a hearty snack.